My name is Yifei Shen. I'll introduce the paper submitted to iMeta. The title is Single-microbe RNA Sequencing Uncovers Unexplored Specialized Metabolic Functions of Keystone Species in the Human Gut. The human gastrointestinal tract harbors a vast and diverse microbial ecosystem that plays a pivotal role in health and various disease states. Gut microbiota are essential for host metabolic processes, including the degradation of complex carbohydrates and proteins. To fully elucidate the ecological and evolutionary significance of these interactions, innovative technologies such as single-microbe RNA sequencing are required to advance gut microbiome research. Through the application of a novel single-microbe RNA sequencing and analytical framework to 3 healthy donors representing distinct enterotypes, this study constructed a comprehensive transcriptional atlas of the human gut microbiome. We systematically investigated functional redundancy and complementarity in central carbon metabolism, analyzing microbial metabolic heterogeneity and covariation patterns at single-microbe resolution. We also demonstrated Megamonas funiformis, a keystone species in Asian populations, can effectively improve mineral absorption through exogenous phytic acid degradation. This study elucidates distinct microbial co-occurrence networks centered around indicator taxa across different enterotypes. The Bacteroides enterotype, ETB, is characterized by bacteroids. The Prevotella enterotype, ETP, is driven by Prevotella, while the Firmicutes enterotype, ETF, is characterized by Firmicutes phi. It is noteworthy that Megamonas has not been reported as a predominant genus in studies of European and American populations, yet it has been consistently detected in both Chinese and Japanese cohorts, suggesting it may represent a signature taxon specific to Asian populations. Employing our previously established microbiome annotation pipeline, we identified 198 bacterial species across the 3 donors and selected 30 core species for functional characterization, integrating single-microbe taxonomic annotation with UMAP revealed distinct cluster-specific associations with particular species. Given the significant enrichment of carbon metabolism-related pathways among species-specific functional marker genes, we conducted an in-depth analysis of central carbon metabolism genes across different species. Building upon observed metabolic gene variations from single-microbe analysis, we systematically investigated metabolic heterogeneity through species-specific metabolic gene enrichment analysis using our newly developed method, metabolism pipeline. The findings suggest that the same species may exhibit distinct metabolic functions across different ecological niches. Single-microbe RNA sequencing of 3 healthy donors revealed significant time-dependent transcriptional changes in gut microbial species, with species-specific marker genes enriched in metabolic pathways and showing distinct diurnal activity patterns, e.g. TCA cycle/glycolysis coactivation in P. copri but negative correlation with M. funiformis. M. funiformis, an Asian-enriched gut species, exhibits donor-specific metabolic heterogeneity and functionally distinct subpopulations, revealed through single-cell clustering and pseudotime trajectory analysis. Key metabolic bifurcations emerged: Cluster 0 dominated central carbon metabolism, Cluster 3 specialized in biofilm-related pathways, and stressed cells showed reduced metabolic activity despite chaperonin overexpression, highlighting niche-driven functional diversification. In vitro experiments demonstrated M. funiformis preferentially metabolizes phytic acid. In vivo validation showed M. funiformis colonization restored serum mineral levels in phytic acid-fed mice by degrading dietary phytic acid and elevating cecal scFAs, suggesting therapeutic potential for phytic acid-induced malnutrition. In summary, this study employed single-microbe RNA sequencing to systematically characterize, with unprecedented resolution, fundamental features of the human gut microbiome, significantly advancing our understanding of interindividual species function relationships. Furthermore, the developed analytical framework provides an efficient strategy to identify keystone functional species with biological and clinical relevance in gut ecosystems.